University. I invite you to the journey, to the wonderful nano world. Some of you may not yet know what does it mean, which dimensions we are talking about. And therefore, just right now, we will explain it to you. So please welcome Aristova Daria with the talk, How Small is Nano? Most important question. You talk. One. Okay, you have me.
Hello everyone, and let me introduce my presentation, How Small is Nano? Before we start talking about nanotechnology, we need to answer the question, how small is nano? And to understand what does exactly word. One means 1 billionth, and nanometer is 1 billionth of a meter. But how we can imagine such a number? This scale bar will give us an idea about size of different objects in our world. And comparing these objects will help us to get an idea about nano. We will start from 1 meter mark. What do we usually measure by a meter scale? Well, for example, human height. On average, the height of a human ranges from 1.5 to almost 2 meters. But when we want to measure some smaller objects, we usually use centimeter scale. For example, mobile phones is something about 12 to 15 centimeters. But there are some smaller objects in our world. For example, insects such as ants. These ants are about 2 millimeters long. So we are now on a millimeter scale, or 1/1000th of a meter. Continuing on to the left of this scale bars, to even smaller objects, we can see human hair. As seen under an optical or light microscope, the width of the human hair is 0.1 millimeters or 100 micrometers. But objects of this size we still can see. So let's move on to objects that are already invisible for us. Such objects are bacteria, such as E. coli. These bacteria have been imaged with an electron microscope, and the bacteria are about 2 millimeters long. Micrometers long. So E. coli bacteria are about 1 million times smaller than a human, because 1 micrometer is 1 millionth of a meter. But viruses are much smaller than bacteria. So our next subject is a reconstructed image of a rhinovirus, which is the cause of the common cold. The width of a rhinovirus is only 30 nanometers. That's 0.03 micrometers, which is more than 3,000 times smaller than the diameter of the human hair. And on the far left side of our scale bar, we have a DNA molecule with its characteristic helical shape. So the width of the DNA helix is about 2 nanometers, which is about 1 billion times smaller than a human. So back to our questions, nano, meaning 1 billionth, describes objects which size are about 100 nanometers and smaller, and which are the left side of our scale bar. Thus, we found out that DNA is 1 billion times smaller than us. Let's go in the other direction and see how big an object would be if it were 1 billion times larger than a human. So the diameter of the sun in our solar system is about 1 billion times larger than a human. So in the other words, nano objects are as much smaller than us as the sun is bigger us. Thank you for your attention.
Wow. Thank you a lot for introducing nano world and even providing the comparisons that we can rely on. So, is there any questions? So, I Nano group, do you know now how small is nano?
Very small.
Very small. Exactly. Okay, thank you a lot. So please welcome our next speaker, Anastasia Skuba, who would told us on the difference between nanoparticles and bulk material. What makes the magic of nanoparticles?
Hello everyone, I'm Anastasia Skuba, and my topic you can see at the screen. Obviously, the main difference between nano and bulk materials is the size, but actually nanoparticles have many other interesting features that we can't explain only by small size. Look at the top picture. We can see how to obtain nanoparticles from bulk material. The volume of first cube is the same as the total volume to all these small cubes. But every division creates a new surface. So at the end, total surface of small cubes are much more bigger than at the start. So we can say that surface volume ratio in nanoparticles is enormous bigger. It is important because how we can see this picture, atoms at the surface of every material different. They can they can interact with other atoms only in one side, and another side is free. It causes more energy of surface atoms. So to balance this energy, atoms in the outer layer of bulk materials are closer each other. But in nanoparticles, there are not so many atoms because they are very, very small. And all the volume of nanoparticles and is very close. So intermolecular interaction in nanoparticles are higher. Another interesting features of nanoparticles, their small, their size comparable to size of free electron path. It means that distance which electron travel from one atom to another. Usually this distance is approximately a few nanometers. So, this the same to the size of nanoparticle. So these reasons cause dramatic change in properties of nanoparticles. This is few examples. First, we can see how nanoparticles in quantum states. I know that fullerenes are obtained in the quantum states, so strange superposition and other things. Because higher surface energy of nanoparticles, they have lower melting temperature but better thermal conductivity. For me, very interesting that chemical properties in nanoparticles change because all interesting chemical actions are in the surface, and catalytic and reactivity properties of nanoparticles are higher than in bulk materials. And at the end, in biology, nanoparticles such as proteins and lipids have can self-assembly. They can spontaneously build new structures, very complex structures. And in medicine, they have various applications. They can be used for drug delivery and for antimicrobial activity. And then I would say that live organisms also can be nanoparticles. For example, viruses, their size compared to the nanoscale, but inside us, actually, DNA and proteins also are nanoparticles.
Is there any questions? Okay. Could you please comment how do you find DNA and protein to be nanoparticle? It can be, the size could be evaluated in nano size?
Yes, how we can see in the previous presentation, the size of DNA is 2 nanometer, protein is viruses at 30 nanometer to 100 nanometer, so you can see they are comparable.
Yeah, but those features which you were talking about, those the molecules, even biological and with nano size, it didn't possess. So the difference of the organization on the nanoscale will make a difference.
Yes.
Thank you. And please welcome our next speaker, Victoria Vasilchenko. Woo! Victoria Vasilchenko will tell us about safety of the nanoparticles, which is a very important topic. Even with all its possibilities and applications, we need to consider and evaluate safety. Yeah? So please.
Hello everyone, my name is Victoria and I'm a PhD student in Kyiv Medical Academy and also junior researcher in Institute of Nephrology. And I with great pleasure continue a topic about nanoparticles and nanotechnologies. What about the biology, biosafety of nanotechnologies? Nanoparticles need to be and could be safety, and it's maybe a conclusion about its safety for each investigation or research. The biological safety of nanoparticles require carefully, deeply, and modern investigation. That is why conclusion about the safety of each nanoparticles needed for update to our research. Different scientific groups makes research of results, and we can see about similar particles that have influence of germ cell populations and such that these scientific groups may obtain their similar results, but for biological safety of each nanoparticles could be and should be provided and further results in further investigation and of research. What about then classifications of nanoparticles? It could be divided in three large groups: polymer, inorganic, and lipid-based nanoparticles. It includes diverse substances, lipid-based nanoparticles, and in their constitution, typical spherical platform comprising at least one of lipid bilayer surrounded at least one intraparticles complement. And as the liquid system, liquid base offer many advantages, including formulation, biocapacity, high bioavailability, a range of biophysical properties that can be controlled to modulate their biological different characteristics. For these reasons, lipid-based nanoparticles most common use of FDA-approved nanomaterials. Polymer nanoparticles consist of nanocapsules, cavities surrounded by polymer membranes, or shell, and nanospheres, solid matrix system. Inorganic nanoparticles consist of inorganic material, for example, gold, iron, silicon, or other. Gold nanoparticles, which are the most well-studied, are used in various clinical research and used like nanospheres or nanostructure. That's all.
Could you please comment a little bit how safety of nanoparticles could be evaluated?
Evaluated in each research, in each scientific books, each nanoparticles can be evaluated for different scientific properties, for example, for structure, and as you can see, different research will give different conclusions, and big research will give conclusions.
Okay. Is there any questions? Okay.
Yeah, why do particles have to be safe? Why do we study biological safety of nanoparticles?
Not all nanoparticles, it's true, would be safe for being used in treatment and in diagnosis, and for all safety, for all biological safety, is not true.
We will have on Wednesday a talk given by Dr. Dumit on hazard and risk assessments of nanoparticles, because this field is very important, and to mainly nanoparticle could be relatively only not safe or toxic, so the dose make a difference, and how to evaluate it and how to classify and register nanoparticles that Dr. Dumit will provide you a very interesting workshop. Okay, so we move forward, and now please welcome Gerasimenko Alexandra. Is here? Are you here with us? Okay, that's interesting. Then we move to Anton Andrenko, I have seen you, who would talk about structural analysis of nanoparticles. Please welcome.
My presentation is a bit tough because I was told it should contain only three pages, so no, I'm for you. I'm sorry. Speaking about analysis of our nanoparticles, currently we have, I would say, like over 20, maybe like over 30 different methods we can use to analyze our nanoparticles. Why do we have so much of them? The answer is that we have way too many parameters to be analyzed, and there is no one unique and universal method to analyze them all. And as it was told before, our nanoparticles they divide into three groups. Right. They may be like inorganic, vesicular, polymeric, and depending on, I mean, they all have like different physical properties. And so, well, choosing our method of analysis, we need to take in mind several parameters first. First of all, like we need to understand what parameter are we studying. Actually, so we can study different physical parameters like size, morphology, porosity, which is our, like property of pores inside the nanoparticle. Also, like surface charge, because our particle may have different surface charge, and also how big it is. Yeah. So we can see like how particles can be different in size, yeah, and also in porosity, in surface charge, and also, well, of course, they can be differently localized within the cell if we're talking about cell, or we can also analyze the binding or aggregation properties, yeah, within our cell or within our solution. Yeah. And also we may use like different study types like in vitro, which is rare, in situ. Yeah. We have different study parameters. Yeah. I'll talk about that before. And well, like our sample can be in different state. Usually we use dry state, but some of them, some of methods may apply to liquid or suspension state or dry state. And if we group our techniques, they can be based on microscopy technique, microscopy like phenomenon, yeah, spectroscopy based, spectrometry based, X-ray based, and magnetic techniques as well. So I'm not going to tell you about all of these groups. I'm going to point out on the basic, maybe most prominent methods I chose. So first of all, we have a large group of microscopy-based methods, and like the most basic one are transmission electron microscopy and scanning microscopy. So these methods, well, they we simply irradiate. We simply use the laser beam on our. We need to have one very thin layer of specimens, and after that we may have a photo like photo projection, yeah, of our specimen. It may look like that. And what's good about that is that we can actually analyze not only shape, but also the like inside of our specimen, yeah, the inside, because our beam it goes through this specimen. On the contrary, and we use confocal microscopy for that. And alternatively, to make it like easier and maybe cheaper in many cases, we may use scanning electron microscopy. So here we simply detect only secondary like electron beam like irradiated from our specimen after after it and goes through this specimen from our glands, our glands. Yes. So in this case, we may have like more visible object, but it lacks penetration depth. So we cannot really analyze what's inside. We can only see what's on the surface. Yeah? And like because our microscopy is is basically basic microscopy techniques are limited by the diffraction limit, which, like, to tell like long story short, it's when we can no longer see nothing but 1 pixel, yeah, from our specimen when it becomes pretty small. So when our specimen is very small, we can only see it as one one random pixel, that's all. So we cannot tell that much information from that one pixel. But we have several techniques to overcome this limitation. So, for example, we can have this donut-like irradiation that sort of catches our excess transmitted light beam, and so what we can see is only this green, like this green point, which is more detailed, and we don't have this unnecessary information in red. And so this is one strategy of super resolution microscopy. Another strategy is to use very weak radiation, which only activates only like one or two of these photoactive fluorescent particles. And then we sort of group our images together. Like first we target this one, then we target this one, target that one, then we group them together, and we have a very nice detailed image without without much of background noise. And so alternatively we can use single molecule fluorescent microscopy, which we can use to study our the functionality of our pores. So our dyes they go through these pores, and so we can sort of analyze how well our pores work, yeah, how well our molecules can transmit through this pores. And finally, we have several techniques which are not based on fluorescence and microscopy. First of all, we have X-ray-based techniques like X-ray diffraction or X-ray crystallization. So speaking about that, we simply use an X-ray and we use it on a well-prepared crystal film, which contains our specimen, and after that we receive a set of beams, and we sort of detect them. We detect their how the whole map. We sort of map them, how they disperse from our specimen, and by doing that we can analyze the structure of our specimen. And we can, for example, we need to know the chemical composition of our specimen, so we make a crystal with it, and then we sort of analyze it. And also we can analyze the radiation, for example, whether our specimen aggregates or not. And we also have, we can analyze our nanoparticles in solution. So we have, for that we have dynamic wire scattering technique and nanoparticle tracking analysis, such that we can even use them in living cells. So we simply irradiate our specimen and see how our nanoparticles behave, how they move, and then we track this movement, and then we analyze it, and then we can analyze the size and speed maybe of these particles. So I think I have already taken all my time provided, so thank you for your attention. If you have any questions, I'll be glad to answer.
Thank you. Very informative presentation. Is there any short question? Just briefly comment, why do we need to know the structure of nanoparticles?
Oh, that's, well, briefly, yeah, so we need to, usually we design nanoparticles, right? And we experiment how to make them. And so usually we need to check whether our methods of producing them is right or wrong, because usually it's very wrong. You need a lot of steps of, you need to modify experiment many times, yeah. And so you need to check how your nanoparticles behave in your test solution or test specimens.
Okay, super. Thank you. So please, now we move forward to the nanomedicine, and it was Angelina Popel. So please come here and explain us what does it mean.
In another time.
Hmm?
In another time.
No. Just a few moments later.
Okay, hello everybody, and it's a great pleasure to be here with all of you at this school. And I would like to present my topic, nanomedicine, the definition and application. So, definition of nanomedicine. Nanomedicine is the monitoring, correction, design, and control of human biological systems at the molecular level and using the developed nanorobots and nanostructures. Simple to say, nanomedicine is the implement of nanotechnologies in medicine. And it can be defined as the use of nanotechnology for medical therapeutics by developing nanoscale agents for the treatment of various kinds of disease. So, how can we implement nanotechnologies in medicine? I will tell it shortly and in general. First of all, diagnostics. Nanomedicine gives doctors a great opportunity to detect disease area and increase the accuracy of diagnostics. And as we know, the more earlier we detect the disease, the more efficient will be the treatment. And nanotechnologies for medical diagnosis and nanoparticles have exhibited great potential for detecting cancerous cells, various viruses, fragments, and specific proteins, antibodies, and etc. Here's what it happens. Nanoparticles are active elements, and they can be used to improve cancer and diabetes therapy, treatment of cardiovascular and neurodegenerative diseases. My colleagues will tell it in more details. So, drugs delivery. Nanoparticles can deliver drugs to specific parts of our body and to do it more efficient as the other drugs. And with the help of nanoparticles, drug delivery can improve the bioavailability of drugs, and we can reduce side effects, so it's better for our patients. And regenerative medicine. By using nanoparticles, we can repair human cells, and we can also repair tissues, bones, and we can get them back to the working state. That's all. Thank you for attention.
Is there any questions? Could you please comment to us whether nanomedicine, is it research or is it already implemented in clinic?
It is, yes, it is some aspects of nanomedicine is implemented in clinic. For example, diagnostics. We can diagnose, we can detect some viruses, some fragments of proteins, of antibodies, yes, but some aspects of nanomedicine, there are a lot of aspects of nanomedicine, they are in process of developing. So nanomedicine, it moves with time.
Yeah, of course. Yeah, but there's a lot of approved already nano drugs coming with the first, for example, doxil was it's a liposomal formulation of doxorubicin, very known anti-cancer drug, was approved in 1995. So don't consider nanomedicine to be on a research stage. It's already implemented, and this year it became even more popular and known due to the vaccines that are also mRNA containing nanoliposomes. Yeah. Super. Thank you a lot. And so we move forward. And next, our speaker is Nikita Polishuk. You are here. Please come to us. Who would further develop the topic of the application of nanotechnology for cancer treatment. So let us welcome.
So, hi everyone, my name is Nikita and my talk is nanotechnology for cancer treatment. It is well known that cancer was the second leading cause of death after heart disease in the world. Cancer is a complex disease which is characterized by several alterations in metabolism, proliferation level, gene regulation, signaling, and cell behavior at all. Despite substantial progress of traditional methods of cancer treatment, each strategy suffers from some limitations. For example, current chemotherapy faces problems such as lack of specificity, cytotoxicity to normal cells, induction of multi-drug resistance tumors. If you're talking about surgery, it's mainly available only at early stage of disease and on certain types of tumors. Radiation not only kills or slows the growth of cancer cells, but it also can affect nearby healthy cells. And despite of all the success and effectiveness of immunotherapy, there is some problem of concomitant autoimmune disease. So none of these therapies are ideal, and each one has its own drawbacks. And however, all these limitations could be overcome by the application of nanotechnology approaches. So, growing interest in applying nanotechnology to cancer is mainly related to its unique feature for drug delivery, diagnosis, and imaging, and also in synthetic vaccine development. Several nanoparticle platforms such as liposomes, albumins, and some micelles have been approved for cancer treatment. If we're talking about cancer diagnostics, in the fight with cancer, early detection is the key factor for successful treatment. And different human drugs and biomedicals, including some peptides, antibodies, and other chemicals, can be used with nanoparticles to label highly specific tumors, which are useful for early detection and screening of cancer cells. Due to poor immunogenicity and limited safety, fewer cancer vaccines have been successful in clinical trials. But nanotechnology can improve delivery systems, improve some pharmacological properties, and provide controlled and localized release of immunomodulatory agents for efficient modulation of specific immune cells. So target delivery is one of the major advantages of nanomaterial-based cancer therapy over free chemical drugs. So enhanced permeability and retention effect is used for passive targeting, while active targeting is achieved by conjugating with antibodies, peptides, aptamers, or other small molecules. So through delicate design and modification of nanodrugs can maintain better specificity, bioavailability, less toxicity to normal tissue, larger loading capacity, longer half-life period in blood, and unique drug release patterns. And this can help to overcome some disadvantages of conventional chemical therapy. So, however, like other therapeutic options, nanotechnology is not completely devoid of toxicities and comes with huge challenges with its use. This means that it can cause some setbacks with their clinical applications. So, only a relatively small number of nanodrugs have been well developed and improved in clinical use with cancer treatment. But all achievements of nanomedicines offer a new opportunities to improve the anti-cancer treatment to find another chink in cancer armor. Thank you.
Thank you a lot. Is there any question? Yeah, please.
How do you think, can we solve the problem of cancer with health and nanotechnology?
I think we couldn't completely cure all types of cancer, but if we're talking about some types of cancer, we can solve some problems with different types of treatments, like chemotherapy and, as for me, and radiotherapy.
So not yet,
Yeah.
but we are on the way.
Yeah.
Okay, so next question.
Yeah, okay. Which mechanism of cancer development are mostly targeted by cancer treatment?
Question about how some nanoparticles can penetrate exactly in cancer cells.
Which mechanism is inhibited?
Inhibit?
Yeah.
I don't pretty understand. No, I don't understand what you mean exactly.
So, I mean, which maybe some signal system or some specific molecule that this drug, for example, target in context of.
Exactly, I don't know about.
Let me help you. Generally, we can answer such question because there's so many different types of nanoparticles, and we need to study each case. For example, if we are talking about drug delivery, then it's more or less in relation to which drug the nanoparticle is delivering. So nanoparticle enhances the efficiency, the concentration, the level of the drug in the tumor tissue. But the mechanism of the action of this particular drug stays the same. So if, for example, doxorubicin is delivered with nanoparticle, as, for example, liposomal formulation doxil, which was mentioned before, the target, intracellular target for the drug stays the same. So it's mainly the DNA synthesis. If we go and study other drug, the mechanism would be of this other drug.
Yeah, so it's some.
Of course, there's so many nanoparticles also interact with signal pathways. So, but it depends on the nanoparticle because each nanoparticle is unique. Yeah, it depends on the physical characterization of the nanoparticle, how it penetrates the cell, which molecular targets it can interact with. So it's a field of board discussions to answer your question, but we can, I hope, during those 5 days, deepen it further.
Yeah, okay, thank you.
Okay, there was one more, and yeah, please.
So with nanopart, I mean, so one thing you didn't compare to was immunology, which was great process, but even of that the nanoparticles isn't the biggest problem the delivery. Like, but I'm not in the field, but isn't that the main problem? Like, to show you nanoparticles only end up in tumor, like they end up in the right cells. So yeah, so we skipped the talk about the lipid-based nanoparticle delivery problem. But can you say something about this?
About lipid or about delivery? It's again.
So is the bigger problem the selection of nanoparticles, or like the right particles, or is the big problem the delivery also, or none of it? And I'm totally wrong. So I'm really not in the field, so I'm asking.
Yeah, thank you a lot for your question.
For me, the biggest problem is targeted delivery to tumor cells.
I would say that it both. The delivery, each case should be considered separately. If we would like to treat a cancer, we develop the nanoparticle-based delivery system for the drug, for example. So we consider the special features of the cancer disease which we would like to target. But cancer is only one application. So in medicine, nanoparticles could be also used for the immunotherapy, for antimicrobial activities, antiviral, and so on, antioxidant also. So each case should be considered what we want to achieve, and then we can think which nanoparticle would suit this aim the best. For example, if we talk about cancer, still the difference of the tumor tissue and normal tissue is considered. As for example, it was mentioned about enhanced permeability effect. So whole nano-sized particles would be accumulated in the tumor tissue much better due to this phenomenon. But also we can target specifically the nanoparticle with a ligand which would target the one unique molecule of interest on the cell surface of cancer, for example. Okay. Yeah. Please.
As far as I understand, if I am right, this is my question. There are also other strategies except for conjugating nanoparticles with, as you've mentioned, specific receptor ligands. These are pH-responsive polymers or temperature-responsive polymers that would, in the tumor tissues, for example, we know that tumors have more acidic environment than the normal tissues, and that would release the drug. And when we talk about temperature, labile proteins and those nano deliveries, we could use electromagnetic fields to cause heating and release the drugs. Perhaps this would add more information.
Thank you. I allow that it drives more discussions. Thank you. So, thank you for your talk. And now we move to the nanotechnology in immunotherapy. Please welcome Anastasia Martynyak.
Hello, my name is Anastasia Martynyak. I am first year student at the Wildau University of Kyiv, Educational Institute of Biology and Medicine. So let's begin. Here, computer. But the phone is not my. So let's begin with what is an immune type of cancer in general. Cancer in general is also known as immuno-oncology, is a form of cancer treatment that uses the power of the body's own immune system to prevent, control, and even eliminate cancer. Immunotherapy can educate immune system to recognize and attack specific cancer cells, boost immune cells to help them eliminate cancer, provide body with additional components to enhance the immune response. Among the most frequently discussed immune therapy styles, we can recognize adaptive cell therapy, cancer vaccines, immunomodulators, oncolytic virus therapy, and targeted antibody. You can ask me a question, where is the nanoparticles? Nanotech in the immune therapy. Let's begin with how does immune response on cancer work? First, it begins with antigen-presenting cells that contain specific antigen. It can be protein from the surface of cancer cell or other types that can show the T cells what exactly it needs to attack. And after this, the T cell have a template and attack these cells if it finds this specific antigen. How nanotech can help this? Antigen-presenting cells usually don't have this cancer antigens because immune system of human recognize the cancer as the same cells as others. So we use different types of nanoparticles to show the disease, show this specific feature of cancer that should be attacked. Naturally derived nanoparticles can be both extracellular, molecular, or peripheral particles, as well as nanoparticles, based nanoparticles, or even molecules. Basically, they present antigen cells and their cycle. If I talk about the most specific classes, we have say what basic. Basically, they are something like varieties of nucleus nanoparticles that again and matrix. Trigger specific pathways that will do general or upregulation of proteins that we can use this technique for regulation of new checkpoints to synthesize specific cytokines that will, for example, attract macrophages or neutrophils to attack the cancer cells. As well, it can be used for T cell engineering to do T cells that target our cancer, and also used to eliminate those tumors. If you'd like to read more about this techniques, you can use the sources. And thank you for your attention.
Thank you a lot. Is there any questions?
Презентацию?
Okay.
Да.
Thank you a lot.
Да.
So let's us welcome Valeria Smalenkovska. Please come to us to tell us about anti-aging applications of nanotechnology.
Копия аппаратной, честно.
Could you introduce yourself to feel the technical?
Hello, everyone. My name is Valeria, and I'm studying in the Shevchenko National University of Kyiv at the Institute of High Technologies. I'm first year master student, and I'm a bioinformatician. So I can start my presentation. Actually, yes. Every person in our planet will face aging, even more. We are aging right now. Moreover, it can lead to age-related diseases such as neurodegenerative, autoimmune, cardiovascular diseases, metabolic disorders, etc. So it is very important to find a tool to slow this process or even to stop it. So, the one of hallmarks of aging is oxidative stress. As an end product of mitochondria functioning, reactive oxygen species can damage our lipids, proteins, nucleic acids, which leads to dysregulation of metabolic pathways, mutations in our genome, and even telomere attrition. Actually, plant-based secondary metabolites are good barriers to beat oxidation in our organism. As you can see on the slide, all these compounds have more than 2 hydroxyl groups by which they can effectively grab these superoxides. Additionally, except this anti-oxidation effect, they prove to have anti-diabetic, anti-cancer, neuroprotective and cardioprotective effects. However, there is a big problem. These molecules have very low oral bioavailability, which is from about 0.1 to 2%, and this number is very small. A good solution for this problem is nanoparticles. And nanoparticles, actually, these molecules which are in these nanoparticles have a bioavailability from 5 to 10 times higher than without them, and that's great. Actually, nanoberberine was used in diabetic mice models, and it is possible to cure this disease. Nanopiperin was used in rheumatoid arthritic mice model, and it's also possible to make things better. Resveratrol is used to cure, not used, but like discovered to cure Parkinson's and Alzheimer's disease. And in this case, it is very promising to cure these diseases because it can go through blood-brain barrier, which is very difficult to go through for different molecules. Another use in nanotechnology, another use of nanotechnology in anti-aging, also connected with another pillar of aging, which is called cellular senescence. Actually, our cell, after a finite number of divisions, goes through this process, which is called healthy limit. And this undergoes cellular cycle arrest and stops dividing and becomes senescent. And senescent cells are more higher, more bigger than normal ones, and they excrete some cytokines, proteins, and factors inside, outside. Yeah. And they even can infect neighboring cells. They can turn them to senescent cells too, or they can kill them. And it can result in tumor growth. It can result in different progeria syndromes, which means when a person ages faster than he or she should. And a good cure of this problem is cellulytics. This is there are compounds which can kill these senescent cells or calm down them. And there is the same problem. It's not available to these cells. We can't send it directly to senescent cell. And actually, Agostini and others designed mesoporous silica nanoparticles, which open their inside only to senescent cells. Actually, housing. We have this mesoporous silica nanoparticle, and she kept by galacto-oligosaccharide. The property of senescent cell is overexpression of beta-galactosidase, this enzyme. And when it enters senescent cell, beta-galactosidase just eat this galacto-oligosaccharide and unpack this cargo inside this nano delivery system. Actually, they didn't show any cellulytics inside this nanoparticle, but they designed a nanoparticle for directed delivery only to senescent cells. That's all. Thank you.
Same questions. Could you briefly comment? You mentioned telomeres and hybrid limit. What is aging on the cellular level for our ID group?
Okay. Hybrid limit. We have on the ends of our chromosomes this short, repetitive, I don't know, like sequences. Yeah, short, repetitive sequences on the ends of our chromosomes, and it works like a calendar of cell divisions. Each time when cell divides, she lost some of telomere ends, and they become shorter and shorter each time. And to prevent shortening of important information in our genome, after in humans, approximate 50 divisions, cell stops dividing, and that's all. Like it stops doing that to prevent some problems with important genetic material, to prevent becoming cancer cell or something else.
Okay, super. Thank you. Please welcome our next speaker, Oksana Chepurna, who will tell us about nanotechnology application for the fluorescence-guided photodynamic therapy.
Thank you for the introduction. I want to start. I want to start from the field where I'm working. Okay. So I'm working in photodynamic therapy. Photodynamic therapy is a minimally invasive method for malignant tumor treatment. It allows us selective illuminate pathological tissue, so you can just inject intravenously photosensitizer. It can accumulate in the tumor, and then you can irradiate it. And also photosensitizer. It is protective agent, so you can use different light for detected fluorescence before treatment, and photosensitizer has several absorption bands, so you can use, for example, blue light for diagnostic, and then red light for. But there is some limitation. You cannot use, you cannot detect deeper tissue than 1 centimeter. So if you want to combine several photo agents, you should use nanotechnology. I'm going to show you, this is our experimental data, and we designed and characterized this polymeric nanoparticles. These nanoparticles allow us to combine near-infrared dye for deeper diagnostic and photosensitizer for treatment. And we want to be sure that there is no energy transfer between photosensitizer and our near-infrared dye. So we just load this agent through nanoparticles. It is called shell nanoparticles. So it also can change the shapes, the diameter during temperature. We can use it for drug delivery, and before that we characterize characterized by these TM images, the TM microscopy, the size, and then we can use this. You can see some structure. This is PNPAM and polystyrene core. So you can use it. You can increase the temperature and it's the same amount of dye. When you increase in the temperature, fluorescence, you can get brighter fluorescence just because the dye can stay close to each other. And also, I want to show you this comparison between actually this work we did with Anya in Shevchenko University. So we just loaded our nanoformulation that contains infrared dye and photosynthesizer, and then we did it free without any nanoparticles. And when we add our nanoparticles and heat it, you can see how bright can we get these images, and also we can detect where is our cells and where the photoagent accumulated. And also we can some dyes actually hydrophobic, and when we just when you want to use this dye, it can form some crystal without nanoparticle, but when we use this nanoformulation and inject intravenously and give these infrared images, you can see small vessels here. And also, I wanted to check why I need these nanoparticles and could this nanoparticle, could this photosynthesizer stay longer? And I injected into mouse in situ, like in tumor, and we wait some time, and because this infrared images and images of photosynthesizer in visual light. So you can see here this nanoparticles, and here just dye without nanoparticles, and after 1 hour and after 60 hours you cannot see any signal, but here still some fluorescence here, and it is for treatment, and this is for diagnostic, and also you can see some signal in tumor. Of course, some nanoparticle can accumulate in the liver, but here there is no signal. And also some video, and you can use this. You can use nanoparticles to understand where your dye or your photosynthesizer or even drug goes which organs. During some time, it accumulates in spleen, in liver, and also in tumor. Thank you.
Is there any question? Please.
I would expect when you make addition of this dye, you block photodynamic effect.
No.
No?
No. Just because the absorption of photosynthesizer about 6 600 nanometers and absorption of dye about 1000 nanometers. And I checked that there is no energy transfer between, and I checked fluorescence, and it didn't decrease. I checked fluorescence of photosynthesizer and of mean dye. It didn't decrease when I put together.
How can you explain it based on energy conservation law? You have excitation energy, and then this energy can come either to photodynamic effect, it means production of radicals, singlet oxygen, and so on. Or it can be transferred to another molecule and come out like this plate.
Just because we use different light, we use this photosynthesizer absorb in red light, and we use red laser for produce signal oxygen,
and here you have done excitation and cerebral.
Yeah.
Oh, okay.
Okay, thank you a lot. Please welcome our next speaker, Olga Yaroshko, who will provide us information about nanotechnology and plant science and even show an example. For that, we give her a few additional minutes. So please welcome.
So, good day everybody, my name is Nano. I work at the Institute of Cell Biology and Genetic Engineering, National Academy of Science, and I want to present you the presentation which is titled Application of Nanotechnology in Plant Sciences. First, we will start from the nanomaterials which used in the plant sciences. We can use the following nanomaterials: carbon-based carbon nanotubes, metals, gold, silver, copper, metal oxide, zinc oxide, copper oxide, titanium oxide, iron oxide, quantum dots, silicon-based mesoporous, thick viscous, polymeric, dendrimers, liposomes, nanogels, synthetic, polyacryl oxide, polypicoline, polyacrylate, polyacrylate, natural, engineered, and phytosan, hybrid, anionic, cationic gel with quantum dots, or metallic nanoparticles at the core. The nanomaterials find interpretation in the following areas: nanobiosensors, which used for plant pathogen detection; metallic and magnetic nanoparticles functionalized with single-stranded oligonucleotides; quantum dots used for detection of phytoplasma. Also, nanoparticles can be used as delivery systems. Liposomes, metallic nanoparticles, metal oxidized fullerenes can be used for controlled release of pesticides and fertilizers. Phytosan nanoparticles can be added to the culture medium. Phytosan has antibacterial properties in tissue culture. Like genetic engineering, my favorite field, the dendrimers are used for gene delivery, the thick viscous are used for plant transformation. The hydrogen nanoparticles can function with DNA. So what effects have the nanomaterials on the plant? Gold and silver accumulate of seedlings germination and vegetative growth. Copper decreases the total biomass, high nanoparticle accumulation, and gene expression in plant tissues. Mesoporous nanoparticles control release of chemicals and nucleic acids. Thick viscous are used for genetic transformation. Hydrogen-based nanoparticles biodegradable and biocompatible materials, which have antimicrobial activity and cause the plant growth stimulation. Dendrimers improve the endosomal escape and delivered DNA. The foreign DNA not degraded then delivered. Liposomes enhance the delivery of encapsulated DNA by membrane fusion, protect the nucleic acids from nuclease activity, and used for cell-specific targeting. So let's look at the methods of delivery of the nanoparticles into the plant. Here, this information in the table, I think it is better to see. Yes. We can use the vivo filtration under vacuum or without vacuum. Injection of nanoparticles with a syringe into the medium of or injection with the syringe without a needle into the tissue of the leaf blade. When using all the methods just listed, nanoparticles are injected through the leaf blade of the plant. The simplest way to deliver the nanoparticles to the plant is simple irrigation of plants with water and nanoparticles. In this variant, the nanoparticles are entered into the plant through the root. The simplest way to deliver the nanoparticles to the plant, more difficult way of delivery is through the tissue culture from the culture medium. For this purpose, tissues of organs or even whole plants are cultivated in the sterile conditions of the nutrient medium in which previously were added nanoparticles. The nanoparticles in this case or separated cells. Thank you, and now I want to show you practical parts. So, as I said that it is possible to use nanoparticles for genetic engineering of plants. I work at the Institute of Genetic Engineering. We usually use as a carrier of DNA agrobacterium. The simplest way to make transgenic plant is to inject the DNA. Here I have simple water, but we can use, usually we should use suspension of bacteria or some other liquid with nanoparticles, and nanoparticles should be covered within a layer of DNA. We'll need syringe without needle. And we will make something like injection. It is not a real injection because we don't use needle. After this manipulation, the water will enter the intercellular space, and this part will be more dark. And if we use nanoparticles with, for example, genes of which cause fluorescence, we then see that when using the source of ultraviolet light. Okay. Also, I take with me a leaf of plant which was already genetically transformed with agrobacterium. This leaf synthesizes at the moment green fluorescence protein. This protein, the genes which are which is called this product, is obtained from the jellyfish, and the synthesis of this product is simple to detect using the source of ultraviolet light. The parts which were transformed will have bright green color. I will show you further on my slide at the end. It will be other ones.
Okay, you can show
You
us this later.
can go here one by one and see the gene plant, please.
Let us do it after the session, please, of the house. When we go to the coffee break, yeah, please come here. Can you read it here, please? And then everyone have opportunity to come and send you a practical example that you can see how it works. Okay. Is there any question? So we have a bit short of time. Let us move to the next speaker. Please welcome Yulia Sklerenko with a talk about nanotechnology in bioengineering.
That's me on the slide. Hello everyone, dear peers and colleagues. It's my great pleasure to be here among you guys. My name is Yulia Sklerenko, and I'm in my second year of master's degree in National University of Kyiv. My given topic for presentation is nanotechnology in bioengineering. It's always hard to start, so I'll start with some definitions to these two scientific branches. Here. So let's start with bioengineering. Probably when a person hears the word engineering, the first thing that pops up that is something that has been uniquely developed and constructed in a specific way. Well, the same thing with bioengineering. Although this something is constructed with biomaterials. And these biomaterials, they could be organic or synthetic. And they also must be biocompatible, which means they also have to promote and support cell's viability. Well, trustworthy sources give more concrete definition, which you can see on the slide of bioengineering. Biological engineering implies knowledge and experience from a number of disciplines and applied sciences. Here you can see chemical engineering, material sciences, physics, chemistry, biology, computer science, and engineering. These are all the disciplines that are applied to the bioengineering. In general, biological engineers attempt to either mimic biological systems to create products or to modify and control biological systems. And bioengineering is used in the design of medical devices, diagnostic equipment, biocompatible materials, renewable energy, ecological engineering, agriculture engineering, a lot of different areas which make our social life better. And here on the slide you can see some of the examples of biological engineering where it's used. Next one. Nanotechnology, on the other hand, is also rapidly emerging technology, which is dealing and developing so-called nanomaterials. And nanomaterials, at least at one dimension, has a size smaller than 100 nanometers. It's aimed to develop systems that can mimic, reinforce, or even create in vivo repair strategies, as nanotechnologies are involved in regenerative medicine via creation of biomaterials and nanostructures with potential for clinical application. And these nanostructures are made from nanometer scale materials, which are called nanoparticles. So a few words about nanoparticles, or how it's also called nanocomposites. Nanocomposites are parts with a size smaller than 100 nanometers, as I already said, and they can be made of a single material type, or it can be a composite of several materials. And for more classification, for more clarification, nanocomposite materials can be categorized as polymer-based and non-polymer-based. And non-polymer-based are inorganic nanoparticles. And furthermore, inorganic particles can be classified into the metal nanocomposites and ceramic nanocomposites. Nanomaterials and nanocomposites are powerful tools for bioengineering applications due to their modifiable structures, which are also have to be biologically safe, compatible, and efficient with the same biological fluids and tissues, mainly because some of them are not detected by our immune system. And it's also clear that nanoparticles are widely used in nanomedicine for diagnostic and therapeutic purposes. So here are some examples. For example, quantum dots. Here they are on this slide. Here. Quantum dots are used for medical imaging application due to its specific optical properties, high quantum yields, photo and chemical stability, and also because of the strong absorption spectrum. Iron oxides and gold nanoparticles, here they are. They also can be used for imaging applications, as they have great absorption spectrum. And my favorite ones are liposomes, hydrogels, and polymeric nanomaterials are great candidates. Here we are. Here. They are great candidates for drug delivery, and they can be used as cargoes and act as Trojan horses. Well, because of the encapsulating the medication, antibiotics, and metallic ions. And in this way, they can deliver medication into the cells at the required dose when it's needed. And the last one is molecular polymers. As a part of matrix material and nanofibers, they can be used for biosensor applications and tissue engineering. And for example, two-dimensional nanostructures are a great scaffold material for in vitro pluripotent stem cells culture, organ patches, and whole organs bioengineering. And also nanomaterials can be applied to improve tissue regeneration, like restoring cartilage function to overcome arthritis, or can be a part of biomaterials for in situ bone reconstruction. Thank you for your attention.
Do we have any questions? Yeah, please.
I'd like to ask, how does the actual drug release happen inside of the cell? Like, how does the carrier detect this, like, arrived destination? It has to release.
When it is in the ribosome, some medication or some antibiotics or ions, like iron and particles, they can be encapsulated into the lipid bilayer, and when coming directly to the cell, it's called endocytosis, and by this processes, the layer of the cell can combine with the B layer of liposome of nanoparticle. This one. But for different nanoparticles, it's a different way of delivery, and it's also a different way how they target the cell and how they enter the cell.
Got it. So you've designed the nanocarrier in this way so that it kind of dissolves when
Yeah. Well, it's not dissolves, it's like builds into the cell. It's how I understand how it works. Maybe some experts can.
With endocytosis, the endocytized, and in endosomes, there could be a more acidic environment, and the nanoparticle can be designed that it's pH sensitive, So in the endosomes inside, after endocytosis, it would be degraded. But here I think he's also some extra stimulation. I'm not sure which power is it. It can be ultrasound, it can be also light, for example, magnetic field. There's a nanoparticle designed to be broken under such a field, and then we apply it targetedly to the tissue of interest where we want to deliver our drug, for example. But there are multiple strategies, of course.
Yeah, or it's also in real light, like Oksana told us.
Oh, exactly.
That's cool, thank you.
Yeah, thank you. So, thank you a lot. Please welcome next speaker, Anastasia Herman, who will talk about nanotechnology and biosensors.
Hello everyone, my name is Anastasia German and I'm a molecular biology master student from National University Kyiv Academy, and today I will talk about nanotechnology in biosensors. So according to EAPAC, biosensors can be defined as a device that use specific biochemical reactions mediated by isolated enzymes, tissues, organelles, or even whole cells to detect chemical compounds, usually by electrical, by optical, thermal, or electrical signals. So in this definition, you can see all the basic principles of biosensors. So let's investigate it more. So we have an analyte that is our substance we want to detect. It's attached to a bioreceptor. A bioreceptor is a molecule that specifically recognizes our analyte, and it can be enzyme, antibody, DNA, microorganism, or even whole organism, or other biological substances that you can see in this list. After this interaction leads to formation of a product, which can be in the form of light, pH change, heat, charge, and mass change. Then, and it's termed as biorecognition. Then we have a transducer that is an element which converts one form of energy to another. Biosensor, it converts a biorecognition event to a measurable signal. Most transducers produce either electrical or optical signals that is proportional to the amount of the product. Following, the signal can be recognized by a computer or even our smartphones that generates an output signal which is understandable for a user. The implementing of nanotechnology will help to reduce the size of biosensor to the nanoscale, which lead to the barrier signal to noise ratio, as well as the possibility to use smaller sample volumes, which mean lower assay costs. Also, we can use biosensors in many areas. In instance, in medicine, it can be laboratory testing, intensive care monitoring, home testing, like widespread blood glucose testing devices that we can use at home. Also, it can be environmental monitoring, biotechnology, food consumption, and even a security system. Most modern biosensors are wearable biosensors that can show the state of your body immediately. Adopting this technology will be relevant for many areas of human life. For example, a tattoo-like patch based on enzyme and parametric sensor connected to the intelligent devices through Bluetooth mode. It will help the police to more accurately determine the blood alcohol concentration of drivers. Also, it can be in a form of gestatin to determine the level of lactates in athlete's body. It is possible to more accurately determine methodology for training and set the intensity of for each specific training session of each specific method. Thank you.
Thank you. Is there any questions? Okay, thank you once more time. And then we move to our last but not least speaker, Bogdan Zhuravai, who will introduce to us nano origami.
So, dear organizers, dear participants, my name is Bogdan, and today I would like to tell you about nano origami. But first, I want to introduce myself and say some words about my scientific interests. So, I'm a third-year bachelor student of the Shevchenko National University of Kyiv at the Institute of High Technologies. There I study biology, and beside this, I work at Wildau Institute of Biochemistry. There at my workplace, I'm exploring the role of the adapter protein RUX95 in cancer organisms, as well as the possibility of its use as a tissue marker of the tumor process. And I consider nanotechnology as a promising area in this case. Especially, I'm interested in its use to targeted drug delivery, influencing by this way the adapter protein that I'm studying. And now I would like to move on to the main topic of my presentation. So the idea of creating nano-origami is to use one single strand of DNA scaffold which will interact with itself by the help of short oligonucleotides. By the word interact, I mean to form some connections. More precisely, holiday junctions. A branch nucleic acid structures that contain 4 double-stranded arms joined together. In fact, you need to mix the scaffold and the short oligonucleotides, and depending on your design, the strands will bend the scaffold in certain places, giving it a certain shape and function. At the picture, you can see the two-dimensional DNA origami structure designed by this principle. Locations where a DNA scaffold shall bend to itself, twist or change its direction, central, are related by hundreds of short oligonucleotides. I have a video for you. Okay. So this was the basic principle how to fold two-dimensional structures. What about three-dimensional structures? To make this, at first we need to somehow modify our scaffold. We can do this by adding or subtracting some DNA bases, thereby making it possible to create holiday junctions exactly where we will need it. Bending that way our DNA, we can literally fold it on the top of itself, like you can see here, the second picture. So basically making the three-dimensional DNA origami is folding the folded two-dimensional structure. Also on the picture you can see different three-dimensional structures that was made by So the most interesting question, where we can use these such structures. At first, I want to show the DNA fluorescent barcodes that we can use to label some components of cells for super resolution microscopy. You can see it here. Just a 6 healthy bundle that has 4 hands of DNA that can be labeled by different fluorescent dyes. The second variant is making some enzyme states. You can see DNA origami with 2 different sites for 2 enzymes. Such cascade can be very essential as it makes possible to do more precise spacing, which can be crucial for certain enzymes. For For example, this goes oxidize and horseradish for oxidize. So on the last picture, you can see two nanorobots that participate in targeted drug delivery. The principle of working with the first nanorobot is that the nano container can be opened only when both aptamers bind to their corresponding targets. And the second nanorobot can be selectively unfolded by nuclein enriched in tumor-associated blood vessels. We had a question, can we cure cancer by using targeted drug delivery of made by nanoparticles? So this second robot allows local exposure of the encapsulated thrombins for intracellular thrombosis that can result in tumor necrosis and inhibition of tumor growth in mice. To conclude, I'd like to say, to repeat that I see this sort of nanotechnology as a very promising area, and especially the possibility of targeted drug delivery. And I hope that they have used this knowledge in their future research. I thank you for your attention, and if you have some questions, we have to answer.
Could you shortly comment, are you working with nanoorganic and will you yes with me?
No, I'm not working with nanoorganic, but I'm really interested in it.
You're going to work on it?
Yes.
Okay, super. Super, I would like to thank you all for, oh, sorry, there was a question, yeah?
Yeah.
Yeah, please come back.
You have mentioned using this technology for fluorescent barcode system. How it is possible to read this code? Because the distance will be below fluorescent, below the detection limit of microscopes.
I think I said that it can be used to super resolution microscopy, and by this it can be read.
Which type of microscope?
Super resolution microscopy.
With scanner. With scanner, to scan the surface, but not with usual microscopy when you
yes,
get the
scan.
Super.
Okay.
Thank you. With that, I would like to thank our Nano Nerds for providing us a solid background to build further the knowledge in the nanotechnology field. You are very welcome to discuss further and to come back to the Nano Nerds in our coffee breaks and other networking opportunities if you have further questions. So thank you.